Welcome to the Deep Dive. Today we are embarking on, well, a truly unique and I think challenging analysis. We're tearing open a stack of sources, detailing a single life, but one's so profoundly split it almost feels like we're talking about two different people who just happen to share the same body.
It is definitely an uncomfortable historical exercise, but I think a necessary one. We're looking at the life and career of Friedrich Röttner. He's born in 1914, died in 1998. And his trajectory, well, it's really a microcosm of the huge political and scientific shifts of the 20th century. It spans, intense ideological fanaticism and then later pure scientific objectivity.
Yeah, the sources really force us to confront this extreme dichotomy in his professional story. It's quite startling. On one hand, you have this active, deep involvement in National Socialist ideology and, frankly, the pseudoscience of eugenics. And then on the other side, a world-leading authority, someone respected globally for work in objective genetics, morphometry, taxonomy of the honeybee.
Right. And the central conflict for us today, the thing we really need to dig into, is methodological. It's about how. How could one person move from participating in a system where biology was twisted for politics, where human racial science was built on these subjective, arbitrary ideas, to then pioneering rigorous data-driven methods that became the gold standard for classifying bee subspecies all over the world.
That's exactly the mission today. We're going to trace his life chronologically. First, looking at those intense political ties in his use and his initial medical career, and then we'll shift to that dramatic scientific rebirth, something that was, it seems, forced on him, but ultimately defined his lasting legacy after 1945.
It really is a remarkable journey from, you could say, the political laboratory to the biological laboratory, and we have to start at the darker end of that journey.
Yeah. Okay, so Röttner was born in Eger, Bohemia, back in 1914. When he started his medical studies at the University of Vienna, he wasn't just, watching the political changes happen. He was actively involved. The sources make it really clear that his early academic path was totally tied up with the rise of National Socialism in Austria.
Yes, and it's important to emphasize this commitment. It actually predates the big political turning points, like the Anschluss. He wasn't just someone who jumped on the bandwagon after the Nazis took over. His political activity started early, and it was pretty aggressive. The records show that from January 1934 right through to the autumn of 1937, he was a member of the SA.
The Sturmabteilung, the SA, the brown shirts. For anyone listening, these were the original paramilitary wing of the Nazi movement. They were deeply involved in political violence, intimidation. And that timeline is critical, isn't it? Because back then, between 34 and 37, the Nazi Party and groups like the SA were actually banned. They were illegal in Austria under the Fatherland Front government.
Exactly. So joining wasn't just a political statement. It was a radical act. It was illegal. And his early involvement wasn't just, say, handing out leaflets. It was considered criminal activity by the Austrian government at the time. The sources actually detail this quite specifically. In 1936, Röttner was judicially convicted for illegal activities related to National Socialism in Austria.
A conviction. And that forced him out of his medical studies for a while.
Yes, temporarily excluded him from university.
Wow. That detail really hits home, doesn't it? It shows the level of his ideological commitment back then. He was willing to risk his professional future, his legal standing, all for this cause. This wasn't some casual affiliation. It was deep-seated conviction years before the Anschluss even happened.
Right. That conviction shows his fervor was, well, stronger than his immediate career goals at that point. It tells us that when the political situation did change dramatically in 1938 with the Anschluss, he was already, vetted, committed, and basically ready to be slotted right into the official state structures.
And the speed of that integration, once the Anschluss happened in March 38, it's pretty stunning. His commitment was formalized almost immediately.
Oh, incredibly fast, with real precision. The sources show he officially joined the SS, the Schutzstaffel, on February 6th, 1938.
February 6th, so weeks before the actual annexation of Austria.
Correct. Weeks before. Then, right after the Anschluss, he applied for NSDAP membership, that's the Nazi Party itself, on May 23rd, 1938. And significantly, not only was his membership granted, but it was backdated, retroactively effective to May 1st of that year.
Which sounds like the regime saying, thanks for your earlier illegal work for the cause.
That's certainly how it reads. It's a signal of appreciation.
So he gets his Doctor of Medicine, the Dr. Med, in 1938. Now he's a documented SS member, a Nazi Party member, and a newly minted physician. In the context of the Third Reich, where does a young, ideologically committed doctor like that go?
Well, he goes straight into the heart of ideological biology and health policy. He transitioned immediately into the field known then as racial science, basically the institutionalized pseudoscience of eugenics. By 1939, Rutner gets a position. He becomes an employee at a really significant and frankly sinister institution, the Biologisches Forschungsinstitut der Führerschule der Deutschen Ärzteschaft.
Okay, let's break down that name because it's a mouthful, but it's crucial. Biologisches Forschungsinstitut, Eugenics Research Institute. Der Führerschule der Deutschen Ärzteschaft of the Leadership School of German Physicians. So this wasn't just some random lab. It sounds foundational to indoctrinating doctors. What exactly did this institute in Aldresa do?
Alt-Resa was a really key node in the whole Nazi system. The institute had basically two main goals, and they were interconnected. First, it conducted research, and I use that term loosely designed to provide a biological justification for the regime's racial policies. Policies that included forced sterilization and ultimately laid groundwork for mass murder programs like the T4 euthanasia program. They did things like measure physical traits, collect genealogies, and try to categorize people based on these arbitrary ideological standards of racial fitness or purity.
So creating the so-called data to prop up state policy, or maybe just fabricating. What was the second goal, the leadership school part?
The second goal was just as important, maybe even more insidious. It was educational and policy-focused. It was set up to train doctors and medical administrators, to turn them into the ideological vanguard, the leaders of the state's health policy. Physicians who trained there weren't just learning medicine, they were being instructed on how to implement laws like the Gesetz zur Verhütung erbkranken Nachwuchs, the law for the prevention of hereditarily diseased offspring. They were being trained to be the key figures carrying out the eugenics program on the ground, making decisions about who should be sterilized, who was deemed unfit.
Good grief. So Rutner was working right at that intersection, political doctrine, medical training, and completely falsified biology. And the sources confirm he was working directly under a major figure in that movement.
Yes, he was under the leadership of Hermann Bohm. Bohm was a well-known, high-ranking eugenicist. And this work, this environment, it defined Rutner's first academic career. A career totally divorced from objective scientific inquiry as we understand it. It was purely instrumental, using the language of biology to achieve political goals. The whole focus was on finding arbitrary characteristics, often just visual things like skull shape, facial features, or maybe a biased interpretation of someone's medical history to divide people up, categorize them, and ultimately condemn whole populations.
We really need to hold on to that idea, arbitrary and qualitative classification, because it's the absolute opposite of what he champions later. But for now, in the late 30s, early 40s, Rutner is established. He's politically convicted, ideologically committed, an SS doctor, deeply embedded in the structures of Nazi eugenics.
Yeah.
It looks like his path is completely set, human medicine, but viewed through this intensely political, ideological lens.
Absolutely. Within that system, it was a path to accelerated success. His background, his commitment, his placement at Alt-Res, it all pointed towards a fast-track career. He was positioned to become a leader in a field dedicated to simplifying human complexity into these crude, politically useful categories.
It paints a very clear and frankly chilling picture of phase one, full ideological integration, not just sort of passive collaboration on the sidelines, deeply involved. Hashtag tag 3. The abrupt historical break, 1945. Okay, let's unpack this massive historical rupture, 1945. If phase one was all about Rutner's total integration into that Nazi ideological system, then phase two, this scientific renaissance we're going to talk about, it could only happen because the entire political and institutional world of phase one was just violently destroyed.
That's exactly right. The end of World War II in 1945 marks a sharp, absolute end to Rutner's first career path. And it was mandatory. It wasn't like he had a change of heart and decided to pivot. It was a direct consequence of Germany's total defeat. His professional context, the ideology that justified his work, the institutions he worked for, they literally ceased to exist almost overnight.
And specifically, how did the Allied denazification process affect him personally, his career?
Well, it resulted in immediate professional consequences. The sources state that in 1945, Rutner was dismissed from university service. He was essentially expelled from the institutions that had defined his professional identity up to that point. The medical and biological paths he'd been on, so closely tied to Alt-Reis, the SS, the eugenics movement, they were completely invalidated by the outcome of the war and the subsequent denazification efforts.
So that dismissal creates a total vacuum for him professionally. It forces a complete radical career change. He was basically barred from the academic and medical fields he'd trained for, precisely because of the heavy political baggage they carried
Yeah.
and his role within them. You can imagine he couldn't really credibly continue in human genetics or even standard clinical medicine in that immediate post-war environment, given his background.
The separation had to be absolute. It was necessary for his professional survival, frankly. He was forced away from that politically charged biology, the subjective measurement of human populations for political reasons, and pushed towards a field that demanded rigorous, verifiable objectivity. That field turned out to be zoology, and specifically apiculture, the study of bees.
That shift, when you lay it out like that, it's just mind-boggling. From applying ideology to human races to applying purely objective data to bee subspecies. It's a move from arguably the most politically contaminated biological endeavor imaginable to one of the most technical and, on the surface, apolitical.
It really is the ultimate pragmatic career transformation, driven entirely by circumstance, by historical necessity. The skills he clearly possessed, things like organizational ability, presumably a sharp scientific mind, deep curiosity, they needed a new outlet, a politically sanitized target. And bees, well, they offered that neutral scientific ground. This shift from the ideological subjectivity of Phase 1 to the quantitative objectivity that would define Phase 2, that's the absolute foundation of his entire second and ultimately much more lasting professional life.
So 1945 acts like this kind of black hole, a forced moment of non-choice that violently ejects him from one orbit and launches him into a completely new one. Right, so the sources show Rutner, this highly educated man but whose education was grounded in pseudoscience, basically unmoored in 1945, but he finds a completely new path. He establishes himself as a zoologist and specifically a Bienenkundler, a bee scientist.
Yes, and the reestablishment of his career starting around 1948, it's quite a story. It involves collaboration, crucial family support, and actually a rapid return to intellectual rigor, but in this new field. The real genesis of his new career was in a place called Lunz am See in Austria. In 1948, he co-founded the Bee Genetics Working Group. This was part of the biological station Lunz.
And he wasn't doing this alone in isolation, right? This restart was bolstered by some established scientific credibility, specifically through his family.
Precisely. That was key. His father, Franz Rutner, was a highly respected scientist in his own right, a limnologist and expert in freshwater biology. And his brother, Hans, was also involved in the Linz station. So this family connection provided a kind of intellectual and institutional shelter for Friedrich. It essentially allowed him to restart his scientific life under the umbrella of established, legitimate researchers in a politically neutral field. Linz became the place where he could dedicate himself to pure, observable, verifiable science, away from the shadows of his past.
He also needed to re-legitimize himself academically, didn't he? His first doctorate was in medicine, but it was deeply tainted by the ideological machinery of the Third Reich.
Yes, absolutely. That second doctorate was essential for his professional credibility in the post-war academic world. He went back and completed a second doctorate, this time receiving a PhD in zoology from the University of Vienna. This move formalized his complete pivot into pure biology. It methodologically distanced him from his eugenics background as much as possible.
Okay, and this is where the conversation really shifts, isn't it? From the historical drama and the political context to some really foundational science. Because Rutner didn't just quietly transition into bee science. He seems to have made an immediate and pretty profound discovery in the very same year, 1948, that he co-founded that Lunz Working Group.
That's right. It's quite remarkable timing. In 1948, Rutner achieved the first documented, verifiable proof of something called polyandry in honeybees. Specifically, he proved that the queen bee mates with multiple drones during her nuptial flight.
Polyandry, multiple mating. Now, that might sound like a technical biological detail, but let's dig into why this was so revolutionary for bee science. What did people believe before Rutner, and how did his finding totally change the game in apiculture?
Well, the prior assumption, and it was a long-held belief in bee science, was basically monandry. The idea was that the queen bee mated only once, or maybe twice, during a very brief flight early in her adult life. This implied that all the offspring in a hive, potentially hundreds of thousands of worker bees, essentially shared the same two parents, the queen, and one single drone father.
Okay, so the hive was thought to be this very genetically uniform, almost like a single huge family with just one dad.
Exactly, and this belief had major implications, both theoretical and practical. Theoretically, it suggested that selective breeding in bees should be relatively simple. You just needed to control the queen and the drone line. But practically, it created a puzzle. Beekeepers knew that hives showed a lot of variation in behavior, in honey production, and disease resistance. Why so much variation if they were also genetically similar? And why didn't this supposed genetic uniformity lead to massive problems with inbreeding depression much faster?
Right, the theory didn't quite match the reality beekeepers were seeing. So Rutner comes along and proves that single mating idea wrong. How did he actually prove polyandry? What was the method he used to show definitively that a queen mates with multiple drones?
It was really a combination of careful observation, classical genetics using visible markers, and later, importantly, refining techniques for artificial insemination which allowed for controlled experiments. Initially, in those early days at Linz, he used morphological differences in color markers. For example, he could crossbreed bees from distinctly different subspecies, say a dark European bee like Apis mellifera with a yellower Italian bee, Apis mellifera ligustica. Then he'd analyze the worker offspring produced by the queen from that cross. If the queen had only mated with one type of drone, all her offspring would show a predictable mix of traits, like intermediate coloration or specific combinations. But what Rutner consistently found, by carefully examining large numbers of worker bees from single hives, was multiple distinct groups of offspring. Some looked like crosses with drone type A, others like crosses with drone type B, and so on. This strongly implied the queen had mated with and stored viable sperm from numerous different drones during her single nuptial flight, sometimes 10, 15, or even more.
10 or 15 distinct fathers represented in the offspring of a single queen within one hive. That completely shatters the old menandry idea.
So, what was the impact of discovering polyandry? How did it change practical beekeeping and the understanding of bee genetics?
Oh, the impact was absolutely fundamental. It basically moved bee genetics out of the realm of guesswork and into modern science. First, it explained the surprisingly high genetic variability within a single honeybee colony. That diversity wasn't coming from multiple queens, it was coming from multiple fathers via the queen. And this diversity, we now understand, is crucial for the colony's resilience, for its fitness. Think about disease resistance.
Ah, right. If a new disease or parasite hits the hive.
Exactly. If the colony was genetically uniform, menandry, a single pathogen could potentially wipe out the entire population if they all shared the same susceptibility. But with polyandry, the hive is composed of multiple subfamilies, each with a different father. It's highly likely that at least some of these subfamilies will possess the genetic traits needed to resist that specific pathogen or stressor. So, polyandry acts as a kind of built-in genetic insurance policy for the colony's survival, a defense mechanism embedded in their social structure in reproductive biology.
Absolutely. It enhances what we call the colony's collective immunity or resilience. That was a huge theoretical insight. Second, Rutner's work and the understanding of polyandry was instrumental in developing modern, effective bee breeding programs. Before this, breeders trying to select for specific traits like gentleness or honey production were often frustrated. Their results were inconsistent. They'd breed a queen they liked to drones from a colony they liked, but the offspring wouldn't reliably show the desired traits.
Because they didn't realize the queen was also mating with potentially a dozen other random drones out on her flight.
Precisely. Once polyandry was understood, Rutner and others could develop much more sophisticated approaches. This included things like controlled mating stations on isolated islands, or perfecting instrumental insemination techniques. With instrumental insemination, you could actually collect sperm from specific selected drones and inseminate the queen in the lab, knowing exactly which fathers contributed to the colony's gene pool. This understanding that you're working with a baseline of high genetic variability due to multiple matings was absolutely transformative for large-scale apiary management and breeding efforts worldwide.
It's just incredible. So within only 3 years of being professionally disgraced and dismissed because of his involvement in ideological pseudoscience, Rutner makes this foundational, objective discovery based on careful observation and genetic principles, and it completely redefines a whole field of biology. This success must have really propelled him back into the scientific mainstream and eventually into leadership roles again.
It absolutely did. And this time, his reputation was built purely on scientific merit, on verifiable results. The rise was relatively swift. By 1965, he accepted a prestigious professorship in zoology at the University of Frankfurt in Germany. And crucially, from 1964 until he retired in 1981, the sources mention his Emeritierung, which is the transition to emeritus status. He served as the director of the Institute for Bee Science in Oberursel near Frankfurt.
Leading the Oberursel Institute, that sounds like it would have made him a central figure, maybe the global hub, for bee genetics and taxonomy research during that period?
It certainly did. Oberursel became world-renowned under his leadership.
And how did he make sure his influence and his methods spread beyond just his own research team? Did he work internationally?
Yes, he was a huge proponent of international collaboration and, very importantly, standardization of research methods. He recognized that bee science, especially taxonomy and genetics, which deal with geographically widespread species, couldn't progress effectively if everyone was working in isolation using different techniques. So in 1970, he was a co-founder of a very influential specialized journal called Apidology. The explicit goal of Apidology was to professionalize bee research globally, to publish high-quality, peer-reviewed work, and implicitly to encourage the adoption of standardized, rigorous methodologies like the ones he was developing. The idea was to ensure that results coming out of, say, his lab in Germany were directly comparable to results from researchers in Austria or France or the US or anywhere else.
Standardization and objectivity again. That theme just keeps coming back, doesn't it?
Sure.
Almost like a complete repudiation of the subjectivity and lack of standards in his earlier professional life.
The contrast is stark. It's impossible to ignore. His leadership at Oberursel, his role in founding Apidology, and his own extensive publications, including his really definitive work, Naturgeschichte der Honigbienen, The Natural History of Honeybees, all cemented his legacy. It was a legacy as a meticulous, rigorous, and deeply objective scientist. He championed a view of the natural world based not on what you wanted to see or what ideology dictated, but purely on what the data demanded you recognize.
Okay, so we've clearly established these two incredibly different phases of Retner's life. Phase 1, immersed in ideological pseudoscience. Phase 2, a leader in objective bee science. But the ultimate intellectual answer, the direct counterpoint to the flawed methods of Phase 1, seems to lie entirely in the methodology he championed and perfected in Phase 2. And that's morphometry. Let's really connect the dots here. How did the arbitrary classifications of eugenics relate to the problems in bee taxonomy? And how did morphometry provide the solution?
Yes, this section really gets to the intellectual core of this whole deep dive, I think. We need to start by understanding the problem Retner was facing when he got seriously into bee taxonomy. And it actually mirrors the fundamental fatal flaw of ideological biology like eugenics. The problem was that traditional classification of honeybee subspecies, what people often called bee races, was notoriously subjective. It lacked rigor.
Can you give us some concrete examples of that subjectivity? What were bee taxonomists actually doing before Retner really pushed morphometry?
Well, they relied very heavily on vague qualitative descriptions, things that are hard to pin down precisely. A taxonomist might describe a particular bee population as being generally darker than another, or maybe more aggressive, or perhaps having a more slender abdomen. But think about those terms. How dark is darker? Color in insects can vary hugely just based on environmental factors like temperature during development or even the age of the specimen. And descriptive terms like slender or aggressive are almost meaningless scientifically unless you have a precise numerical standard for comparison. Aggression is behavioral, hard to quantify consistently. Slenderness is relative. Often, the classification was based simply on where the bees were collected geographically, maybe combined with one or two obvious visual traits like the overall color pattern.
That sounds disturbingly familiar. It sounds methodologically very similar to the approach used in eugenics and racial biology, doesn't it? You find one or maybe two easily visible, easily defined traits like skin color or head shape, or in this case, bee color, and then you use that as an arbitrary basis to define a distinct, supposedly fixed group or race.
The methodological parallel is striking. It's inescapable, really. Both systems, the old bee taxonomy and racial pseudoscience, sought to simplify complex biological reality. They tried to categorize living things based on insufficient evidence, on selective observation, and often on criteria deeply biased by preconceived notions. The crucial need in bee taxonomy, just as the critical need was and is in studying human population variation, was to move decisively away from subjective judgments based on just looking at something. The need was to move towards objective quantitative data, numbers, measurements, statistics.
And Rutner's solution, the bedrock of his work, especially during his time leading the Oberursel Institute, was the massive development and application of morphometry. Let's explain exactly what that is.
Okay, so morphometry, at its core, is the quantitative analysis of biological form. It's about measuring shape and structure precisely. Rutner didn't invent morphometry. The basic idea was around, but he really refined it, standardized it, and applied it systematically to bees on an unprecedented scale. He essentially weaponized it for taxonomy. So instead of just saying a bee from population A looks smaller than a bee from population B, you get out your microscope and your calipers or later digital imaging tools. You precisely measured the length of the proboscis, the tongue, to the nearest hundredth of a millimeter. You measure the exact width of a specific abdominal plate, say the third tergite. And crucially, especially for bees, you measure the angles formed by the veins in the wings. Wing venation patterns turned out to be very informative. This process moves classification completely away from the observer's subjective impression, it looks darker, and into the unambiguous, verifiable language of numbers.
And this wasn't just measuring one or two things, right? The sources emphasize that Rutner and his team standardized the measurement of dozens of different characteristics on each individual bee specimen. How extensive was this data collection effort?
Oh, it was an absolutely massive, painstaking undertaking. At the Ober-Ursel Institute, Rutner implemented a standardized protocol that involved measuring, I believe, somewhere between 20 and 30 precise morphological characteristics for every single bee specimen they analyzed.
20 to 30 measurements per bee.
Yes. And not just on a few bees from one hive. He insisted on collecting large samples, dozens, sometimes hundreds of bees from many different locations across the entire geographical range of a suspected subspecies. This was critical for understanding the natural range of variation within a population. He refused to define a race based on a few bees from one spot. He wanted to capture the whole picture. And all this extensive data, millions of data points, was meticulously organized and stored in what became an incredibly valuable resource, the comprehensive bee database at Ober-Ursel.
Why was it so vital to measure, say, 20 or 30 different traits? Why not just find the single most obvious difference, like maybe wing length, and rely on that? Why embrace all that complexity?
Because nature, especially when you're dealing with subspecies or populations that gradually blend into one another geographically, rarely draws sharp, clean lines based on just one characteristic. And this is exactly where the old subjective methods completely failed. If you take two genuinely different subspecies of honeybees, say one from the Alps and one from Italy, their characteristics will often overlap considerably if you only look at one measurement at a time. For instance, the average proboscis length of subspecies A might be, let's say, 6.4 millimeters, while subspecies B averages 6.3 millimeters. There's overlap. Some bees in A will have shorter tongues than some bees in B. That single measurement on its own is statistically pretty weak for reliably separating individuals or even populations.
Okay, I see. But when you measure 20 or 30 traits simultaneously on each bee,
That's when the true statistical fingerprint, the unique profile of each group, starts to emerge. Rutner and his team utilized sophisticated multivariate statistical techniques. Methods like principal component analysis, PCA, or discriminant function analysis. These are statistical tools designed specifically to handle many variables at once. They can analyze the data from all 20 or 30 measurements simultaneously. So while the two subspecies might overlap quite a bit on wing length alone, and also overlap on proboscis length alone, and overlap on tergite width alone, when the statistics look at the combined pattern of all those measurements together, the overall multidimensional shape in statistical space, the groups often cluster out much more distinctly.
that makes perfect sense. It's like facial recognition software. It's not just looking at eye color or nose shape, but the combined relationship of dozens of points.
Mhm.
That's the technological, or rather the methodological antidote to subjectivity. The old subjective approach picks one easy, maybe misleading difference. The morphometric approach uses mathematical rigor to define a group by its total, complex, objective signature.
Absolutely, that's a great analogy. This rigorous quantitative methodology allowed Rutner and his successors to objectively differentiate honeybee subspecies that the traditional qualitative methods had either failed to separate properly, or had mistakenly lumped together, or had even incorrectly split apart based on trivial variations. He demonstrated, through massive amounts of data, that true biological classification, if it's to be meaningful and stable, must embrace complexity. It has to use powerful statistical methods to draw lines based on real underlying patterns, not just on what looks convenient or obvious to the naked eye.
This work effectively placed the entire taxonomy of the honeybee, Apis mellifera, onto a much more solid, unshakable scientific foundation than it had ever been on before. His legacy, in this sense, essentially prevents or should prevent any future attempts to impose simplistic, arbitrary, or non-objective criteria onto bee classification. The data standards is just too high now.
And that is the profound intellectual irony we keep circling back to. The very skills that Rutner applied with such discipline and efficiency in his second career. The meticulous organization, the dedication to collecting comprehensive data, the insistence on institutional rigor and standardization were utterly devoid of ideological contamination when applied to apiculture. He shifted from a world where biased, selective data was used to simplify and categorize humans for political control, to a world where overwhelming, objective, multivariate data was used to reveal the true, complex, and beautifully nuanced structure of honeybee populations across continents.
It really feels like the ultimate testament to the necessity and maybe the eventual resilience of empirical truth in science.
Indeed. His work in morphometry established, maybe unintentionally but powerfully, that the only acceptable scientific method for defining biological groups, whether you call them races or subspecies or populations, is one that systematically eliminates subjective judgment through rigorous, data-driven statistical analysis.
So let's try to pull this all together. What does this incredibly complex story mean for us, for you, the listener, as we look back on this extraordinary, deeply divided life? We've traced this figure, Friedrich Rutner, who spent his youth and his entire first professional career in active, even criminal support of a political ideology. An ideology that prized arbitrary racial distinctions, simplification for control, and biological pseudoscience. That career path was abruptly, violently terminated by history. And then, the same man emerges to become a global leader in a scientific field that is defined by the meticulous statistical rejection of arbitrary distinctions, defined by objective complexity.
The extreme dichotomy, the split in Rutner's life, it really does offer a remarkable, if unsettling, case study. It shows how external forces, how forced change can lead an individual to champion the very kind of scientific objectivity that the ideological systems they once served actively sought to destroy or corrupt. He transitioned fundamentally from using biological research as a tool to justify political doctrine to using biological research purely to understand the intricate structure and variability of the natural world.
Just think about the contrasting goals of the two key institutions in his life. Alt-race in Phase 1 aimed to simplify human beings into rigid, politically useful boxes. Aryan, non-Aryan, subhuman, hereditarily sick, healthy based on selective, qualitative, often fabricated data. Then you have Oberursel in Phase 2. It aimed to measure dozens of data points on thousands upon thousands of individual bees using complex statistics just to accurately delineate two closely related overlapping bee populations. It demanded complexity in the pursuit of truth.
The shift is just profound. It demonstrates a complete 180-degree realignment of methodological priorities, of what constitutes a valid scientific evidence. While his early career sought to bend biology to fit a predetermined ideology, his entire later career was spent ensuring that biological classification remained subservient only to empirical evidence and rigorous statistical analysis.
And this brings us to the essential, maybe provocative final thought for you to consider as we wrap up. The very techniques that Rutner helped pioneer and champion in his second life, the objective quantitative taxonomy using morphometrics, things like meticulous multivariate measurement, comprehensive databases, advanced statistical analysis, these were precisely the kinds of powerful methodological tools that would have been necessary to scientifically discredit and dismantle the pseudoscience of racial biology in the human context as well, if only they had been applied rigorously and without ideological bias from the start.
It's a powerful point. His life unintentionally demonstrates that methodological rigor, that commitment to objective data, is perhaps the final essential bulwark against the corruption of science by ideology. If the same intellect, the same organizational capacity, could first serve an ideological system that valued arbitrary distinctions, and then later establish a purely objective scientific framework that valued quantitative rigor above all else, what does that ultimately tell you about the power and maybe the inherent ethical neutrality of the scientific method itself? Is the method separate from the person wielding it?
Does the robust empirical method somehow redeem the individual practitioner later in life? Or does it simply demonstrate the absolute, enduring necessity of objective truth, regardless of the scientist's personal history or motivations? That's a really powerful question, I think, about the responsibility that comes with scientific knowledge and capability. Something for you to mull over until our next deep dive.